So welcome back, students. Am I audible to you all?
Yes, sir.
Okay.
Yes, sir.
So today we have to check what we have done first. Assignment part. Okay, so the classmate section all of you have uploaded, and I have created one more assignment part that is for your lab report, that is your agrogel electrophoresis. So that assignment you have to submit by tomorrow 12 night. Okay, so before moving to our next practical, so any one of you would like to ask any question, any doubts regarding the DNA generation process? Any query?
Excuse me, sir.
Yes.
Sir, what will happen to the DNA fragment if the current will put on for a longer period of time?
See, where you are asking in agrogel electrophoresis?
Yes, sir.
Okay, so what is the role of current in electrophoresis? What is the role of current?
Now I have a meeting and I'm going.
Sorry, sir, can you repeat?
I'm asking what is the role of current in the hydrogel electrophoresis?
Sir, to move the molecules.
To move the molecules. Okay. So if you keep on continuing that turn on the currents, then your molecules keep on moving, and they will leave the gel. They will come out of the gel. They will come to the solution of the buffer that you are having in your buffer tank.
Okay, sir.
So just I will have you. I will just sharing a video. So using that video, I will explain. So what is going to happen when you keep your electricity on or your voltage on? Just wait for a moment. Same video I will share you that I have shown you earlier, I think, or some new maybe. Okay, so I'm sharing your video, so hope it will be audible to and you are also able to see it. I will give commentary in between, so that is going to answer you, Nathan, what you are asking. So is video visible to you? Are you able to see the video?
Yes, sir. Yes, sir. Yes, sir.
So as you can see that here the two edges are there. One is this edge and second is the another edge, and here you have placed your form, and this gel, this tray you are placing into your buffer tank, and at the place of where the well are there you are going to load your DNA. So then when you load your DNA within this well, your DNA start moving to your gel. Okay, so I'm playing it, then you can see.
And the techniques that we study from very technical user perspective.
So here you are seeing that the buffer are flooded over your gel. So gel is completely dipped inside your buffer. Okay. So this is the thing how you need to do. Is the voice is also audible to you? Voice of video is also audible?
So we are essentially we are not interested.
Is voice of video is also audible?
We are being very
So not really.
Sorry?
So it's not audible.
Okay, just is it audible now?
Yes, sir, it's audible now.
Yes, sir.
Okay, so as I mentioned that it is well flooded. So now thing is loading your DNA, and DNA you are placing within the gel. So to place within the gel, you have made the holes that you are calling that as a well, and in the well you are placing your sample. And your sample you are placed with the loading die. You can see that how you have to load it. So every well is getting one sample. And xylene, cyanol, and bronze will create these two dye I have already mentioned you. So xylene, cyanol is run around 5,000, let us say. And another one, orange G also you can use. So depending on what composition you want to do, you can take. This is loading dye, and loaded. Just place over the well. Because simply thing is that as I mentioned, these S's are there, so DNA is going to run within this gel. It is going to migrate from the negative toward the positive one. So as if you keep on, if you do not monitor the gel status, how it is running, so in end, what is going to happen? Your gel DNA will come out of the gel. So for that purpose, whether DNA is coming still inside the gel, you are using that tracker dye. So tracker, that xylene, cyanol, bronze that we are saying. So they are going to track the position of your DNA. So till you feel that your DNA is within your gel, you keep on running. So this is a ladder. That ladder will tell you the size of your DNA. What size of it is, just approximation. A small size DNA you have loaded, and you know that at which location which is going to be there. So this is how this really loading is happening at the gel. This is a black to red migration. Black is going to be negative, red is going to be positive. Voltage, you have to fix it. If you keep voltage high, then the temperature is going to be rise. That may lead to the melting of your agar gel. So like if it start running, it start entering within your gel. You can see how it is migrating. See, now it's leave the well and enter inside the gel, and it keep on moving. With time drive, it keep on moving. It keep on moving. So if you keep it on running, then definitely it will leave the gel. And when it leave the gel, you will not change the fate. So when as you can see here in this case, so here you just allow to run the gel. You can say this blue dye is just in between, or you can say wells are at this point position, and total gel is of this much length, and it's around 60% you have run. And if you found that you need more, you want to run it more, then you further need to run it more. But you can't allow your DNA this dye to come out. So if you keep on running, if you forget after adding your DNA to the gel, and you move to for the tea and so on, and there you start chit chatting, and you forgot that to turn off your gel, then your DNA will lose, your gel will lose the DNA. So here you can see that how your bands are going to be appear. So first lane you have added the ladder, the last lane also you have added the ladder. Your sample was there. In one well you just added just only the dye, not other than that dye. So that is to tell you that band that you are getting is from your sample, not from the dye or some other contaminants. So more intensity, more bright is a more amount, less intensity is going to be the lesser amount. So in the imager you have to see in a black and white mode, not in the colored one. So your imagers are black and white. So wherever the intensity of light are there, then you will see a color, white color band, and where no DNA is there, no fluorescence is coming out, you will see a dark background. So here you can see that your wells, just a moment.
वो शायद खरीदना ये भाई पता ना ऐसे फोन कराना फोन करते रहे टी शर्ट्स कम बहुत कुछ होगा मिलके नहीं देखना ना नहीं होगा ना नहीं होगा
Okay, so this is how gel is done. You can see some well are also a bit bright in color. So that may be because of that your some remnant DNA may be there. In that case, that's why wells are also showing intensity. Or that may be because of edges also show some reflection. So because of that also you are seeing the brightness in the well. Otherwise, if DNA is going to be there, then brightness is going to be a bit more. So this is the way how to find the size. So you can see that both left and right side there are the ladder. So here these bands of the ladder are of known size. It is provided by a company, so they know, they tell you the what size is there, what is the smallest band size, and then how these bands are going to be increased. So like simply if you say the smallest band is a 500 base pair, then 1000, 2000, 3000, so on. So in this way you can find the approximate size. So here is not the actual size. Approximation we can only do in this case because you are just comparing by a standard one. So sometime it may be a bit upper position or a bit lower position, then you can't say clearly what is the actual size, but approximately you can say that it is around that much. So you can't say that it is 2502 over there, then simply you can say it is around 3500. So this is ethylene bromide that I mentioned you. This is a fluorescent dye that you are adding to your solution. It came like 10 mg per ml is a solution that coming. It already also come by in the powder form. So if it is going to powder form, then you have to dissolve into your buffer or water, and as it is in directly you can add to your gel. So the thing is that one more thing with ethylene bromide is that the direction of migration of ethylene bromide is reverse. So if your DNA is moving from negative to positive, so then your ethylene bromide is going to move from the positive to the negative. So with that, the both are moving in the reverse direction. So then because of that migration, ethylene bromide is going to entangle within your DNA. So this ethylene bromide you need to add into the gel itself while you are casting your gel. So this is another dye that you can also use. The name of this dye is Cyber Green. Cyber Green 1. There's also a fluorescent dye. Your ethylene bromide is fluorescing orange. As its name is green, it will give you the green fluorescence. And it is a bit less carcinogenic as compared to your ethylene bromide.
There are important safety issues to consider when using bromide. It is a mutagen that intercalates and binds the DNA of a PCR product. It is best to assume that everything to do with bone gels is contaminated with bromide. This includes gel equipment and anything used to prepare gels, including the sink, cabinets, microwave, and also the camera system. Always wear gloves in the gel preparation area and wear a lab coat to protect your clothes. Do not touch the rest of the lab with contaminated gloves. Remove gloves immediately when finished and wash your hands.
This is a simpler thing. So what happen if you are running it overrun it, and why it is coming out? So it's a bit simple explanation. And any other thing that you would like to ask? Nehan, is it clear to you now? Nehal?
Yes, sir. Yes, sir. It's clear.
Okay, so any other question
No, sir.
other than Nehal? Okay, so now we can move to our next spectacle, and that is going to be restriction digestion. And when we say the restriction digestion, simply means you are going to utilize the restriction enzyme. And about restriction enzyme, we have dedicated many of our theory classes. What restriction enzymes are, how they are going to work, what their specificity is, what their different types, what are their requirements, and also the property that I mentioned, that is your star activity, if it is going to change the buffer condition or the pH, that we are going to do. So as they are enzyme, so enzyme we generally get in a form of unit, and definition of what is the definition of unit you can in biochemistry class might be your teacher told you, so how we are going to what unit mean. So when we come to the our restriction nuclease, so 1 unit of enzyme will cut 1 microgram of DNA. That's simply you can say that when we are saying hard DNA, like you said, when you talk about the unit, in the case of neurosubstrate, you might, I think you might define in the amount of your enzyme which is able to produce a catalysis change in 1 mole of uh, your substrate. When it controls 1 mole of substrate to the product, that amount of enzyme we are going to call it as the in a unit time. So unit time is also needed. But here we are saying 1 unit of enzyme. That is that amount of enzyme that will produce cut that is going to cut 1 microgram of DNA in 50 microreaction in 1 hour. This is a description of unit of enzyme in case of restriction endonuclease. So so how much of DNA you want to cut, depending upon that your requirement is going to be, and what volume you are going to make. That also we have a question that if you want to go for a large volume of reaction, then you may need a large amount of enzyme, a bit large amount of enzyme also. But the main thing is about your amount of your DNA. How much DNA you want to restrict digest. So if you want to restrict digest 5 micrograms of DNA, then you need 5 microliter of 5 unit of enzyme, so that you can get yield in 1 hour. And second, one more thing is there about your restriction digestion, that you want to go for a partial digestion or a complete digestion. So here partial digestion and complete digestion, what do you mean? So partial means, like simply if you take an example, your DNA may have 5 restriction sites. Like if you say 1000 base pair long DNA you are having, and every interval of 100 bases, you are having one restriction site. It means it is going to have like 5 restriction site, but 100 and 200, 300, 400, 500. So in one way or second or another thing, we say that at 200, 400, 600, 700, and sorry 600, 800, then we are going to get 4. So one more you can reduce at any other place. So we say just for restriction sites at 200, 400, 600, and 800. So when these 4 action sites are there, so how your restriction link can work in this case? So just for I think I'm just taking an example of your whiteboard. Just a moment. Okay, so is whiteboard visible to you all?
Yes, sir.
Okay, so simple as I mentioned, like this is your DNA, and this DNA is of 1000 base pair long. So what we are saying, uh, 4 restriction sites are there. One, like this is 0, then this may be 200. Second is there, this is 400, and this is going to be the third one. This is the 600. This is going to be the 800. So now what can happen in this case? So two possibilities are there. One is a complete digestion. When we say the complete digestion means your all the sites are cut down. When all the sites are cut down, that means you are going to get 1, 2, 3, 4, and 5. So the 5 cuts are there. So all are at a equidistance. So you are going to get a single band in your DNA. That band is going to be of 200 base pair. This is complete digestion. Second, we are saying partially digest. Partial means it's not digesting complete. So means in one it may digest at this location, another it may digest at this location, in third it may digest at this location, fourth may digest at this location. So a combination of the locations might be there, like one band may be of this size, another may be of this size, then maybe of this size, or maybe of this size whole, or you may say that this one may be there. So different combinations may form. It means when you are having a DNA simple, so like we say that like this is your gel you are running here, and in this gel these are your wells. Okay, so when you are having intact DNA, that intact DNA of this type. And when we mention when it's digest complete, this is of 1001. So when digest complete, we are going to have only abstract of 200, the single band. But when the digestion is a partial, so that means you are not going to get, you don't know where it is going to occur. So you want all type of the, all size of the bands. So in that case, you are not going to see a distinct band, you will see a smear type of. Okay, this is going to be your smear. So means your bands of different lengths are there in it. So depending upon if the fragment size is going to be, like here equidistant we mentioned, at equidistant they are located. That's why you see a different combination for 4 raised to power whatever the site number probability you will say. So all the combination size are going to be there, and in other case, complete digestion, one or two sites are going to be there. So this is the thing that you mentioned, that complete or the partial digestion. Now you may ask that, sir, when we should go for a complete digestion and when we should go for a partial digestion. So the thing is that when you do not know, means you want your gene intact, but you don't know that whether within your gene your restriction site is there or not. Or you don't want all fragments should be small, so you want in between intermediate fragments also, because your gene of interest may be there. So for that you are not going for complete digestion, you need to go for a partial digestion. So in the partial digestion, what possibilities are there? You may got your gene intact. So the depending, but if you know that you want to know the complete profile, what fragments you are going to get when the complete digestion is going to occur, in that case you need to go for a complete digestion, like RAPD, that RFLP, restriction fragment and polymorphism when you are talking about. Or in that case you need to digest it. For the southern blotting that we talked about, so in case of southern blotting also you need to go for a complete digestion. But if you experimental requirement calls for a partial digestion, then you need to go for partial digestion. Okay, so then come to how to perform the experiments, and what precaution we should take, what are going to our requirement to perform this experiment, what information, what background information you need. The background information that we need is, the first you should be aware about what restriction enzyme you are going to use. And when we are seeing the restriction enzyme, that simply means recognition site you must be aware about. If I'm calling that EcoR1 I'm going to use, then I must be aware about that EcoR1 is recognizing what restriction site. This is the first basic information that we need. This enzyme is recognizing this thing. So whether it is going to produce any cut within your DNA or not, so for that you must check whether the restriction sequences are there or not. So, so for that, how to choose your restriction enzyme? There are various sites which provide you which restriction sequence is there within your sequence, which enzyme is going to recognize. Just a moment. So there are the various websites are there that you can use to find the which recognition sequence are there in it, which enzymes you can use to produce a cut. Because knowing the restriction site present within your gene, you need to know not just to produce a cut, but also many times happen you do not want to produce a cut. Like as I mentioned that if the restriction sequence is present within your gene, and you want to isolate your gene from your genome. So in that case, you want to use your restriction enzyme. So in that case, you want to use those restriction enzymes which are there, whose recognition sequence are there in the flanking region. Okay, flanking region means from the left end or the right end of your gene, not within your gene. If those recognition sequences are going to be within your gene, then if you are using those restriction enzyme, just a moment, इसकी आ रही भाई पानी पीना पड़ेगा. Okay, so as I was saying that, so for that I will share you. Have you used those restriction sites or in your previous semester? Anyone have told you the finding the restriction sites? No. Okay. So just a moment, I will share you those sites where you can get which are going to help you in finding the restriction site within your genome. One simple site is the nab cutter. Just a moment, I will share you those sites. Okay, so here you can see the first requirement is going to be we must have some sequence, some DNA sequence. So DNA sequence you can get from the site is there where your sequences are available. That this is a NCBI nucleotide. Okay, so here I give a search for hemoglobin. So here I'm going to get the sequence of hemoglobin. Different formats are there, so you can like it's a sequence from, from one to it's a very long sequence, so you can choose what of sequence you need, what of length. So from here you can pick your sequence, required sequence that you are having. Like I picked up to this 1020-21. Just copy it, and then I mentioned different sites are there, different they are providing different tools. Like one is the NAB cutter, so it is a tool that provided by the New England Biolabs. So here you can see that here you have to paste your sequence. Just copy and paste it here, and format you need the FASTA format, so bioinformatics it might tell you, and then what type of the enzyme you want to check. Lab enzyme, so this portal different enzymes are there. So what type of your sequence is linear or circular that you can feed it, and plasmid vector, any standard sequence if you want to, or if you don't have that, leave it. Okay, minimum aura flame display, that's also a thing. Okay, so then you have to submit. When you submit it, so NavCutter will provide you all the details where different sites are going to be located. So you can see that from this first one, so this at which location different enzymes are there. So LW1, DNP1, MOB, BST, BAMH1, and South 3A, DNP1, they are located at this point, and then AF3. So all these are this all map is there. So where so this sequence that you are having, this sequence have all these enzymes. The origin site for all these enzymes. Okay. So on the basis you can choose where they are located. So like you can see this NSIL, NSI1. It is the covalent site is ATGC80 and is located at 519, 519 location is located. So if you choose this enzyme, then it is going to produce one cut, and some enzymes which are going to be there for which there are more than two sites are going to be there. So just check which enzymes are there. So yeah, you can see BTS. No, this is BST. BST. This is BST. The V3 is there. Okay, so no two sides for any enzyme is there. So, but you can find here that anyone having more than two sites. So this is the way to check. And similarly, another tool is there that's called the GenScript. So GenScript also provide you to study you have to do simple thing as we have done earlier. Just you need sequence, just copy your sequence and paste in the next one, that is GenScript, and submit. Okay, so it's submitted your sequence, the sequence, so now they are the sites. You can see where their sites are. So they are going to be there. So there are a long list of enzymes whose site are going to be there. So here you can see that I'm asking about any enzyme which having two sites. So you can say that there are the enzymes. This is the ACLW1. It has two recognition sites in it. That's one, and that is a 10 and 4. Then if you follow the L1, L1 4 to 229, to 231 1, 502 to 504 second, 805, 866. So if you treat with the L1, then you are going to get 5 fragments because 4 resting sites are there for this enzyme. Similarly, if you talk about the ALW1, it have 2, and others are having 1. So this 3 are there for BF2, 2 are there for BL. So these are different enzymes, they are different about number of enzyme sites. So on this basis, you can choose which enzyme is suitable to you. So I simply can say that when we are talking about uh, our most important enzyme, that EcoR1. So eco R1 site is not there in it. Is it there or not? Yeah, there is no site of eco R1. Eco is there, but not eco R1 is not there. So this way you can choose your enzyme, so which are not producing cut within your gene and may produce cut outside your gene. So that is going to help you to get results. Similarly, another tool is also there. This is restriction mapper is the name of the tool. Restrictionmapper.org. It's not opening, so you can open it in your portal also. So nap cutter, GenScript, so these are the tools that you can utilize for producing restriction, for checking which restriction site is going to be there. Sorry, in your gene. Sorry. So in this way, you can use your tool to find your restriction location, restriction site where it is going to produce cut. Similarly, if any sequencing is being provided to you, one more simpler way is there. So like in this way, you need to do. Some ways are there where you don't need any tool. So what is that simple way? Just copy it and paste in your Word file in a gap-free way. Then what do if you know that this is a recognition sequence for this enzyme, like then give a command Control F, and like if you know that your sequence, your enzyme that you are having, it has recognizing AGGTA. So you can see this sequence is going to have 55 AGGTA. So in this way you can check where AGGTA are there, like one more A I am also going. Now if you are recognizing the 8 sequence now, you can say the sequence probability of finding it and is going to come down. So only 8 sequences are there. This is a simple way. If you are aware about what recognition site is there for your enzyme, just simply by just Control F also you can find how many location this sequence is there within your gene, in your sequence. Otherwise, you can go for the tools that are available online, that is a nap cutter or GenScript or gene mapper, restriction mapper you can use. Okay, so we will continue with our restriction digestion in our next session also. So now please turn on your camera to record your attendance. Just a moment. Okay, when I call your name, please turn on your camera. Demetrius. Demetrius.
Yes, sir.
Tridev. 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 Manaswini.
Present,
Adish.
sir.
Present,
Adish.
sir. Present, sir.
Yuvraj. Yuvraj.
Present, sir.
Yuvraj, turn on your camera. Sanjana.
Present, sir.
Nehal.
Present, sir.
Atoiba.
Yes, sir.
Devanshi.
Present, sir.
Nandini.
Present, sir.
Kshitij.
Present, sir.
Janvi.
Present, sir.
Janvi. Shivani.
Present, sir.
Shakti. 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 Rupal. Rupal.
Present, sir.
Sahiba.
Present, sir.
Ishita.
Present, sir.
Present, sir.
Namrata,
Present, sir.
Tanvi,
Present, sir.
Yukta,
Present, sir.
Ayushi,
Present, sir.
Vivek, 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 okay, so 2 students are absent, 3 are absent, I think. Tridev is absent, and Shakti is absent, and Vivek is absent. Shakti, Tridev, Vivek. Shakti, Tridev, Vivek. Okay, so thank you, students. Thank you all. Stay safe, stay happy. We'll join back after 5 minutes.